Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It is our great pleasure today to have Dr. Jinjack Wang to give us BrainMap talk. Dr. Jinjack Wang is Senior Research Physician and the Clinical Director of Laboratory of Neuroimaging at the National Institute of Health, NIH, and National Institute on Alcohol Abuse and Alcoholism is Intramural Research Program in Bethesda, Maryland. Prior to his current appointment, he was a professor of radiology at Stony Brook University in Long Island, New York, and senior scientist and chairman of medical department at Brookhaven National Laboratory, Long Island, New York, and held a joint appointment as professor of psychiatry at Mount Sinai School of Medicine in New York City. His research focused on application of positron emission tomography, PET, and MRI to study various brain disorders. He is interested in using PET to study neuropsychiatric mechanisms and manifestation of alcoholism, drug addiction, attention deficit and hyperactivity disorder, obesity, and eating disorder in humans and animal models. Using PET, his first reported similarity of human brain circuits disruption in substance use disorder and in obesity. Currently, he uses PET and MRI to study brain dopaminergic signaling in opioid use disorder. His other interest is using MRI to assess gut and brain interaction after bariatric surgery. His studies were supported by the Department of Energy, BNL, NIH, State University of New York, and pharmacological companies as a principal investigator or co-investigator. He has published more than 360 original research articles and 55 review articles and 20 book chapters. I would like to remind our audience to address your question by raising your hand or in the Q&A box at the end of the talk. And thank you very much for giving us talk today, Dr. Jinjack Wang, and the stage is yours.
Thank you, Chiong, to invite me to give a talk to the outstanding imaging center. My talk is focused on obesity. I've been studying obesity since the 1990s, and I'm going to share my research with you using that brain imaging method. Okay. Obesity has been kind of like a pandemic in the United States since that 80s, and that it getting worse and worse. That you can see that the proportion of the severe obesity is getting more, and this is not only in the United States. Actually, in fact, it also prevalent in over the world, especially in the developed country. And U.S. is considered home to the highest population of severe obesity people, and China is second. However, for the number-wise, that China is the number one, especially in that most obese nation already overtaken the U.S. And so my research is that initially focused on that population in the United States. Lately, my research also worked with my colleagues in China. And obesity is the problem that developed a systemic disorder. You can see in this slide that all over the body affected by that the obesity. And that one of the issue we are particularly interested is especially in the psychosocial issues. Recently, we published a review article in Molecular Psychiatry showing that using that the MRI will allow us to understand that how the obesity affect the brain, and that obviously associated with abnormalities of the frontal mesolimbic circuits and the impaired cognitive function. And using the MRI will allow us to know from the behavior all the way to how it affects our brain in terms of that the functions such as homeostasis, motivations, and reward, learning memories, and then also allow us to deep inside the brain, not only in the cortex, all the way into the subcortical structures, and that this allow us to know more about the brain and how the obesity impact to the brain functions. And that our brain is not only made of that the neurons, also these develop into the network. And that because all these that each neurons are causing by the impact from that obesity, and then that the networks that also affect it, and these that alternation into decision-making network, deformal network, and that from these that stimulus recognition networks. And this alternation may contribute to their higher craving for food and greater perception of hunger and lack of the control of overeating. So I will summarize that what I'm going to talk today. Since obesity associated with brain changes include that strong brain response to food cues, and more food craving, a lack of control of overeating, compulsive overeating, and increase the perception of hunger, impair memory of how much you eat. And the overeating behavior in people with obesity is very similar to compulsive drug use in people with a substance use disorder. And compulsive behavior in obesity and the substance use disorder, and their dopamine is the one underlying all these issues. Lastly, I'm going to talk about how the body weight reduction after the bariatric surgery altered that the brain emotion and that behavior in people with obesity. And this would lead us to learn more about that how the brain can be recovered after the this traumatic limitation to the food intake, and that also led us to learn about that neuroplasticity. So way back to late 80s and early 90s, we start to study that substance use disorders such as cocaine, alcohol, and later on we start the methamphetamine abuse. These drug and alcohol, they have the very common phenomenon is that they have the decrease at brain dopamine D2 receptor availability compared to the age control subjects. And that they, as we see from that PET study using that carbon-11 rockopride, and in the late 90s, we even studied that the people with that severe obese, that means that the people with a body mass index more than 35, and that and we found that the people with severe obese also have decreased dopamine D2 receptor availability, make us to find the similarity between these substance use. And then we want to know how that the brain, why the brain dopamine system is involved in that the brain dopamine system by using the food stimulation method, by showing the person with a real food, the food they prefer, and then we give them a list of the food items they them to choose, and then we give them we show them in front of the PET scanner, and then we inject the carbon-11 rockopride, and then that compared to the non-food items in the other day of the study. And then we see how the brain responds to this food stimulation by seeing and with the smelling and talking, and then without eating. And we found that compared to the neutral stimulation, that the subject have lower the dopamine D2 receptor binding. That means that this is the area, means that the release of the dopamine during that food stimulation. And subsequently, we use that FDG. This is that would allow us to how the brain use that the glucose, and is also help us to understand the brain function during using the same food stimulation method. And we found that it activates many areas of the brain. The particular interest is in that areas such as orbitofrontal cortex. And when the person, they express their feeling of the hunger, we find that that area has greater to increase that dopamine, the glucose uptake, FDG uptake in the orbitofrontal cortex. And so this we know that orbitofrontal cortex is in part regulated by the dopamine activity. So this is the key brain region control the planning behavior. And so it determines the presence and the probability of the food. The enhance of this orbitofrontal cortex activation by food stimulation are likely to reflect the downstream dopaminergic effect, and likely to participate in the dopamine involved in the drive for food consumption. And then that we further to understand that if the food stimulation can enhance that the brain use of the glucose, and that means that then if we ask the person to inhibit themselves when they receive the food stimulation by their attempting to distract themselves, even the food in front of them, the food they like the most in front of them, and they taste it, they smell it. However, they straight to suppress themselves, to ignore my stimulation to them. How would that be? And we found that it's very interesting is that we found that the person, they suppress their desire to eat, and that they also have the report, they really say that I don't feel hungry at all. And that and you can see this is the time sequence, and when they we give them the food stimulation, and then that and then we inject that FDG here, then we scan somewhere around here, and for 20 minutes. And then that we found that the brain showing that many areas of the brain region, they decrease activations compared to that food stimulation state. And those are the areas related to food cravings, such as anterior cingulate regions, and striatums, orbitofrontal cortex, amygdala, hippocampal area, and the cerebellum. And that so the suppression of this region involved with that regulation of the satiety and motivation to eat suggests this is a mechanism by which attempting cognitive inhibition decrease that desire for food. So that is that when we use that the PET will allow us to use the real food. However, when we want to do MRI, we find that there are some difficulty in a facility to do that. So I work with a team in China by using that food cue. We give them the cue of that high caloric food pictures, and low caloric food picture, and that and neutral controls. And those are non-food items. And then we compare with that normal weight, overweight, and that people with obesity, and that means that more than 30 BMI. And we found that by using that resting state and see their response to these food cues. And we found that obese group and had both a greater baseline activity and food cue induced activation. They in both that overweight and normal weight group. And you can see that. And then that later on, we also to try to see which area of the brain regions has most of that changes that during that racing state and that response to the food cue. And actually, we find that the most traumatic area is in the area in the left hippocampal, in the hippocampal amygdala area. And that we found that obese group, both in that racing state of the in the hippocampal amygdala activation and the high caloric food cue induced response will correlate with that body mass index. However, we did not observe that in that normal body weight or that people with overweight. And we also try to see how would that react, the relationship between the body mass index, the activation in that hippocampus amygdala area, and the high caloric food cue. We found this that relation between that body mass index and the left hippocampal amygdala area response to high caloric food cue can be mediated by their racing activity. And follow that, as I mentioned to you, that we have the attempt, the inhibition, cognitive inhibition, and we did it in using that PET. And so we would like to know if that the last study was done in a person with that non-obese, and we want to know if we compare to the obese subject and how much the difference between that obese compared to non-obese. And so this is the study. We did it in that a group of 16 obese subjects compared to the one I showed you before. And we found that non-obese has a significant increase at baseline to food stimulation, and the decrease from that food stimulation to that attempt inhibition. However, the obese subject, they has a significant decrease from that baseline. And you can see that in the orange bar to the food stimulation, and increase in that in the food stimulation to in the glucose in metabolic activity in the same area. However, there is a one area particular different is that in that anterior cingulate cortex, BA32, or we call pregenuine that ACC, PGACC, that that is a very interesting area that is related to that food craving, inhibition of the food craving, and that the non-obese subject that has a less desire for food during the attempt, the cognitive inhibition was associated with less activation in that BA32 in the ACC. And this is consistent with non-obese subjects showing less activation in this cluster during attempt inhibition than the food stimulation. So that means that obese subjects, they need to use more energy to separate this area in order for them to deal evenly with all these attempts. And we know that obesity, why they see the food and then that they have cannot control themselves, they to use a lot of activity, brain activity to inhibit it, even they want to inhibit themselves because they have a higher impulsivity. So we by using another methodology being used that on the study of the substance use disorder called delayed discounting task. And that so we modify the content of the delayed discounting by ask them to say that would you rather to have that 20 Chinese dollar now, or you rather to have the 100 Chinese dollar in 2 weeks? And then that all these tests that I know you guys are expert in this too. So I just brief to teach you how we modify this to tailor to that our study subjects. And so we would allow us to test to see their inhibition control. And that so we found that the subject with obesity prefer to eat right away rather than waiting. For them, the eating is a bigger reward than that exercise is. So they have the higher impulsivity. And so we see a lot of the brain changes that our differences in that the person with obesity, and that we know that how we eat is affected the brain, what we brain tell us. Could it be also our guts tell us? And so that we know that whatever the food we eat, eventually will go into the guts and then absorb into the body. So it go into the stomach and then to our intestine, eventually absorbed, and then it coming feedback. And so this is a very complex that process. So brain is not complete separate from our body. There is a gut and brain interaction. And that so when we eat and then that we feel full, so that means that the brain will send a signal. So brain sends a signal by many means. That, for example, the body has a signal go through the vagus nerve, so send that the inhibition process to know that how much we eat, and then when we eat, and then this signal will send the so-called satiety signal to the brain stem and to that nucleus tractors, the solitarius, and then from there and into that subcortical area and into the cortex, and it becomes a story to tell us. And this is the gastric vagus effect signal increase the firing, respond to ism, also respond to the mechanism pressure from the injection nutrients. And then that, and then it respond to that the food induced release a lot of these that brain gut peptides such as cholecystokinin, ghrelin, or glucagon like peptide 1 to the vagus nerve, or they can penetrate to that blood-brain barrier system. And so we did a study that by using the balloon into the stomach to increase that the pressure to the stomach, just like when we ate and then expand the stomach. And usually that the stomach can be spent roughly around 700 cc on average. And so we, instead of giving 700 cc maximum, that sometimes after that people start to feel pain. Well, no, we just want to give the person feel something, so we give the balloon with a core setting around 500 cc to let the person feel something. And when we give that increase the warm setting into the balloon, and we found that actually it suppresses on many areas of the brain compared to when the person is empty in the zero balloon. And then that, and we found that the area decreased activation is in that area such as the cingulate and striatum, and that also we found that decrease in the orbitofrontal cortex, hippocampus, and the cerebellum. It looks like very similar to I showed you before, right? And then that also increased that activation in the insula and amygdala. As we know that the insula, and especially in the posterior insula, is the area where the vagus signal will send to that VTX, and then hit to that the brain is one of the areas receive the signal is in the posterior insula, and amygdala is again like the area with that a very early detection area will let us know. So we found that also measure that the grading before and after the person received that the balloon. We found that the subject had a greater increase of the peripheral grading level after the gastric distension, also had a greater activation in the left amygdala. And then let us understand that how brain, when they respond to the mechanical expansion, and the brain will respond in this area. And so the expansion, again, it will touch, and then the signal will go to the vagus nerve, right? So there was an Italian surgeon, and he found that actually if you can manipulate that the vagus nerve, and then so it might let the brain understand the stomach is full, and it's such that the person would not eat. And so he developed the method by giving the stimulator, is a pacemaker, into the laser curvature where that vagus nerve go through, and from here to the top, right? So he put this pacemaker to the stimulation with different degree of the pulse into the brain. And then so at the time, one of my colleagues that she got a job to work with this a company who doing the clinical trial. So with her arrangement that we allowed to study this the subject who received that implantable gastric stimulators. And then so we have the person shift over the United States to that New York, Long Island, and so we half of the subject they come in, turn off for 2 weeks, and then and come to us, and the other one that we scan them during that the stimulation, and then we turn off after the study, and so that we have these. Then we sum them up. So, and we found that when we compare to the stomach is being stimulated by these gas stimulators, and then we found that the gastric stimulation, and compared to no stimulation that means off, and that we found that this gastric stimulation activate the brain in that hippocampus, and then that also activate that striatums and the orbitofrontal cortex. And so these are the same area activated during that the drug craving in subject with the substance use disorder, supporting the similarity between that compulsive overeating and drug addiction. And so we are very interested to study all this the gut and brain interaction. So the next step that I try to find a collaborator in the United States to study that the result of bariatric surgery, how use that as a model to help us to understand how the brain recover after the surgery, right? So we know that once you start decrease that eating, and then the body, and then the person would decrease that the body weight, and then the person not eating, people thinking that, well, maybe because they don't eat, and that's why body weight decrease. And then because they are surgeon, they may not thinking of the brain actually contribute a lot to this the process. As that neuroimaging investigator, that we are very interested to understand how the brain contribute to this whole process. And actually also will allow us to understand how the body weight decrease, and the person they don't want to eat, and they have a different kind of the feeding process, and their behavior and their psychological changes. It will give us an opportunity to understand our brain, how plastic our brain is. And we know that the brain, the stomach has lesser curvature and the greater curvature, and these are the area. The greater curvature actually is the area is the region produce ghrelin. Ghrelin is only a peripheral hormone or peptide. They promote the eating process. The rest of the 12 hormone, including the insulin, that leptins, or the peptide, or CCK, or GLP-1, they all suppress our eating process. And that and then ghrelin stimulated the dopamine release from that ventral tegmental area, and that so this is the only hormone outside the brain that make us feel hungry. And that in our study, we found that bariatric surgery has right now the most common two. One is that so-called sleeve gastrectomy that remove that larger curvature. The other one is called bypass surgery. They remove all this area, and they only have pouch, and then directly link to that terminal ileums. So bypass, and then that is more dramatic, but there are more side effects. But is considered the most traumatic bariatric surgery. So in this surgery, we found that the sleeve gastrectomy, we found that cause a reduction of that body mass index. That you can see this is pre-surgery. You can body index decrease, and then the excessive weight also decrease gradually. Decrease almost to 74% after 1 year. And then also the person, when we did that FUQ study, they also decreased their craving for high caloric foods. And then that a month later after the surgery, we want to understand how the brain changes even in 1 month. It's not only because of their they cannot eat that much anymore. However, we found that their brain change. One of the area, the most dramatic is actually in the hippocampus. And that as you see that before the surgery, we found that the resting state function activity is that the brain activity increased with that higher body mass index. And after the surgery, that in the hippocampus actually significantly decreased. And also we found that the brain activity decrease in the hippocampus also decrease with the decreases of that the grading. And also when we measure that the Hamilton anxiety rating scale, the person, the subject also claim they have less anxiety. And also we found that after a month after the surgery, actually enhanced that the brain activity in the posterior cingulate gyrus. And that is that associated with that the greater increase of the in this area also decrease that anxiety. And then we also did that delayed discounting test, as I showed you before, and then see how the surgery affect. Actually, before the surgery in orange line compared to the green line, this is normal weight person, that increase, they have a much higher discounting rate, and that after the surgery in the blue line, it actually is less. And that so we see that there is a difference in that a choice for a smaller immediate reward in that left that dorsolateral prefrontal cortex and also right cortex, and that also difference in that hard choice. At least we find that in the left dorsolateral prefrontal cortex and left medial prefrontal cortex. However, it increased that activity, related activity in that in the insula bilaterally, and these are all the relative that related to their the brain behaviors, for example, such as their cognitive controls and their feeling of the hunger and their increase their disinhibition. And so this that the subject with obesity decrease in passivity after that the surgery was associated with the normalization of the activation in region involved in the executive control, reward evaluation, and their eating behavior. And then 6 months later, we also did a study to see how their response to the food cure. And that we found that the most dramatically is that we found it decrease their activation in the dorsolateral prefrontal, and then that the changes in the dorsolateral prefrontal also related to that food craving. The decrease of this area decreased their food craving. And so when we did use that DLPFC as a C to see which area has most of the enhanced their connections, and actually turned out is in that the ventral ACC is almost the area that we asked the obese person to suppress their desire for eating, and then actually the surgery is seems that make that this group of subjects much easier to suppress their food craving. And a year later, we want to see how that the local density and that brain activity density, and then we found that actually the area pop up is that in Athinoula. You are so surprised that this is an area we rarely see because of such a small regions. But 12 months after surgery, we found that it's become very predominant, the activity we can identify. And the Athinoula is actually, people describe that is area called anti-reward, or the area involved with the negative emotionality, and that and so we use that this as a C to see how the structural connectivities by doing the DTI study, and found that these actually increase that their connectivities, structural connectivity in area related a lot of hedonic eating process, such as in the orbitofrontal cortex, superior frontal gyrus, related to this Athinoula, and also to that amygdala I mentioned before. And another one is that we want to see how would that affect that the area such as that hypothalamus, the area with hemostasis. And very interesting is that also related to their eating behavior. For example, this is that the area when we compare to the persons that the Dutch eating behavior questionnaire, the one pop up is that related to their external eating. So the person with the increase of their connectivity between the Athinoula and that hypothalamus actually also decrease their external eating scale. That means that when they're eating in response to external related cues. Another one also very interesting is that they also decrease their Hamilton depression scales. And so the strong connection in the brain also have the person, the strong break when you are depressed. And then that we also try to see how that Athinoula functional connections and to their after the surgery. And we found that after the surgery, a year later, and that the gray matter volume in that Athinoula actually increased the size, and then that the increase of the size also related to their decrease of their body mass index. Another one is that they also increase their functional connectivity, especially into the left insula, the area, posterior insula, as I mentioned to you, related to our gut functions connection area. And then that this resting state connectivity also in this area also related to decrease, also related to their decreased body mass index. And they also decrease also related to that their food addiction scales, and also decrease their emotional eating. So that increase the Athinoula volume and reduce the connection between the Athinoula and the insular connectivity contribute to that body weight loss, and for the person to have the less emotional or mindless eating. And not all the surgery, especially in the gastrectomy, are so lossy. And then so you can see this publication since from 2016 to the last report on the 2021. That from this publication showing that yes, they have a very great after the 10 to 15 years of the follow-up. They have some of them have the very good success rate with an excess of weight loss of the 75%. To the last report, only 31.5%. So the last one, this is published from the Austria. Austria that they found that actually the other group of the person almost that they had to receive another surgery. As I mentioned to you, the bypass surgery is more traumatic surgery doing the bypass. And that 49% of the person need to do that. So what happened? Why some people, they are doing so well? I mean, not the surgeon, I mean that the patients, but some people, they cannot. It must be some reasons, right? So we are thinking maybe there are people, maybe their genetically component is different from the other one. So maybe we know that obesity involves so many genes, metabolism, related to our eating behaviors and all those kind of things. So we choose that the one because is the name, you can see that so-called fat mass and obesity associated gene. And we use that to MRI to see how the people with this carry this gene, especially the people with their variant. And one of these SNPs, I call the IS 9939609, has that variant called A. And we know that this gene is regulated at the dopaminergic midbrain circuitry. And so we found that a year later, the people, they have that this A variant. This is normal, and this is the variant. The A variant that they have less decrease of the body mass index. And you can see in this that red line. And that we also found that in these the subject, the baseline activity, as we know that usually after a month later, they increase their brain activity in the posterior cingulate cortex, right? Instead of they are not much like the rest of the group. So this that FTO A variant allele carriers has less weight loss and the low baseline brain activity in the self-regulation, the region after the surgery. So that we also want to see how these variants are different in terms of their brain response to the food cue. And we found that from this, we found that in the person carrier that area with a less weight loss, and you can see that. And compared to the TT, had a greater response to the food cues in the area such as that related to a reward or appetite control. And you can see that anterior insula, DLPFC and DMPFC after the surgery. So there are stories keep going on because that we are still following up the subject, and the other project we are working on and we haven't published is that and we still seeing is because we want to see another portion of the gut and brain interaction. As I show you that we have allowed these hormones interact with that the brain such that affect that the brain activities, and then also affect that our behaviors. But the other big portion that is that our friends in our guts, those are our microbiomes. These microbiomes actually they work very hard for us, and these microbiomes they would also involved with our digestions, and then that after, and they do a lot of fermentations, and they produce a lot of these metabolites and the short chain fatty acids, and they also produce a lot of these the neurotransmitters. And so this is the next step we are working on. We have some data, but we are not ready to show you that. And so this is that work in progress. And then that we want to compare all these line of that process in our body, and so then that we can link to see how this the surgery, in addition to we see the hormone affected our brain, and how this the metabolites that produced by these microbiotas, and which microbiota the one they contribute the most after the surgery, and then how it affects to our brain. And so this is that. Okay, so I want to conclude my talk is that obesity is associated with brain changes that make it harder to control craving and overeating. Those brain changes can be reversed through the lifestyle change, including exercise and mindful eating. And also, in this study, show that more research on the gut-brain interaction can treat and prevent overeating. And that I would like to thank everybody for your attention, and then that I also thank my collaborators. And this study was done in the Brookhaven National Laboratory before Department of Energy decided to shut down their clinical medical research in Long Island, and and also my colleagues in that NIH, especially Dr. Nolobokov, and my colleagues in China, the imaging expert and the surgical experts. Thank you very much.